So I am really excited to be here. It's one of the first talks I've given in person since lockdown, so you have to bear with me if I'm a bit rusty. But it's really lovely to be here in person and to see all your faces in the audience. So thank you very much for having me. So yeah, I want to do we're going to do a bit of a whirlwind tour through the whole Phanerozoic tonight. So we're going to go from 500 million years ago right through to present day and potentially even a little bit into the future. Looking in particular at the role of mycorrhizal symbioses in evolution of plants, of soils, and the climate. So I always like to start by kind of making the point that we often get told that Earth is a blue planet, when actually it's not. It's a green planet, right? Plants are the dominant kingdom on Earth, and they make up the vast majority of carbon present on the terrestrial land surfaces. So of the terrestrial land surfaces, if we think, well, yeah, forests on their own, so that's just trees and forests, they make up 31% of Earth's surfaces, and that includes all oceanic surfaces as well. And of the 550 gigatons of carbon present on Earth, plants are actually responsible for 450 gigatons of it. Now, it's really hard to visualize that amount of carbon. It's a bit of an abstract idea, but so I kind of like to equate it to elephants, right? So it's the same mass of carbon that plants are responsible as 50 billion African elephants. If you can imagine that volume and that mass of carbon, that is all plants. But it wasn't always like this. If we get in a time machine and go back 500 million years, there were no plants on Earth's terrestrial surfaces. And actually, it may have looked a bit more like this, with exposed rock, barren land surfaces with no terrestrial life, save for maybe a biocrust made up of lichens potentially, or bacteria. But that wasn't the case across all the land surfaces. At the water's edge, so the marginal areas, there were kind of cyanobacterial mats and bacterial and algal films that gave some color and life to the land surfaces. So you can see here that the green indicates there's some photosynthesis going on. And this was kind of the first sort of almost terrestrial photosynthesis. When plants did make the move onto land, however, things started getting a bit more exciting. So this is an artist's impression of what Rhynie would have looked like. So Rhynie is a site in Aberdeenshire. And this is what it may have looked like when the first plants started coming onto the land. And this is around 410 million years ago. And what you can also see in this artist's impression are the towering structures in the background, which are actually probably fungal. Okay, so those are prototaxites. So how do we know that it looked anything like that at that time? Well, it's actually thanks to the environmental conditions at Rhynie during that time, which were kind of special. So if you go forward to today, this is what Rhynie looks like now, and it's really unremarkable. Okay, but actually, back when back in the 400 million years ago, the conditions were totally different, and it was a very sort of geologically active area with lots of hot springs, highly silicified waters, which were very conducive to forming incredible fossils. So if you scratch the surface of Rhynie, you reveal rocks, and in those rocks are the most amazing fossils. Okay, and the preserving power of the waters of Rhynie, thanks to the environmental conditions at that time, was so great that you actually have preservation of early plants almost intact. So this is an early plant, and you can see the bifurcating branches and the spore-bearing structures held at the end there. But it gets even better than that. So if you look in some of the fossils, you can actually take microscopic sections or sections of the fossils themselves to look under the microscope. So this is a section taken from the axis of this plant here, which is called Agolophyton major. And this is a transverse section taken from this axis here, which kind of snaked along the ground. And when you look under the microscope, it's incredible. You can make out individual cells of the fat fossil. What's perhaps even more exciting is this dark smudgy band around the center here, around the cortex. And when you look inside of that, you can zoom in on your microscope. You can actually even see these sorts of structures here, okay, which are clearly different to the plant structures, but they kind of bear a lot of similarity to these structures that we see in modern day plant root cells. And these structures are formed by fungi that live in the soil and form close partnerships with land plant roots, which we call mycorrhizas. So what are mycorrhizas? I'm sure I'm in the right place, but mycorrhizas are the term we give to those partnerships formed between certain groups of soil fungi and plant roots. Okay, and it's through mycorrhizas that plants are able to get access to a much greater volume of the soil and thus more nutrients, which the mycorrhizal fungi are able to scavenge for, and they transfer those nutrients to the plant root in exchange for organic carbon fixed through photosynthesis. So in this way, mycorrhizal symbiosis is usually thought of as being a mutualistic relationship between the plant and the fungus. So in the soil, the fungi kind of live like this as spores, which respond to chemical signals coming out of the roots, which makes the spores germinate, and then the hyphae grow out towards the root. So mycorrhizal fungi are really excellent at getting mineral nutrients out of rocks. So they can even, they can secrete acids and actually through that, they actually liberate mineral ions from the rocks themselves in the soil. So they kind of provide plants with an alternative pathway to getting nutrients out of the soil. So it's not just about expanding the area the plant can forage over, but also getting access to different types of nutrients in the soil. So this is what they look like under a microscope in modern, this is in wheat. And you can see here, these highly branched structures are what we call arbuscules. And through these structures, the fungus gives up mineral nutrients. And then in these structures here, these are big vesicles, and we think those are like the carbon storage structures. They tend to be quite full of lipids and other carbon-rich molecules. So we normally consider mycorrhizal symbiosis, and in particular, the symbiosis that involves those arbuscule-producing fungi, which we call arbuscular mycorrhizal symbioses, and I'm going to refer to those as AM from now on because it's a bit of a mouthful. We normally consider them to be mutualistic in that the plant gains nutrients in return for carbon, and they sit on this end of the scale. But in reality, this symbiosis actually lies on a continuum that runs from full mutualism, where both parties benefit from the relationship, through to parasitism. So this plant here has even gone so far as to actually lose its chlorophyll and relies instead entirely on its fungal partners to get all of its nutrients and carbon. And so we class that as a mycoheterotroph, parasitic on its fungus. You can also lie somewhere in between. So orchids, for example, they need a fungal partner to germinate from their dust-like seeds. The fungus actually gets into carbon from either the leaf litter surrounding it or from other plants it's got associations with. And then the seed can germinate. And then when it becomes photosynthetic and produces its first green leaves, it can pay back that carbon debt to its fungal partner. And this plant here, Ophioglossum vulgatum, it has tongue fern. It oscillates between being mycoheterotrophic during the season it's trophophore, this green photosynthetic leaf is below ground. It becomes mycoheterotrophic, and then when it starts photosynthesizing again, it switches and then becomes more mutualistic. There's lots of different factors can affect where any single partnership lies on this continuum. And that includes things like competition, the genetic diversity and compatibility of the partners, the nutrient status of the soil, the type of soil, the disturbance, and also atmospheric CO2. So just to illustrate how flexible this symbiosis, I just wanted to do I wanted to go through a quick example one of my PhD students had done recently, looking at what happens if you change the availability of carbon to the mycorrhizal symbiont. So he used aphids as a carbon sort of competitor in that aphids feed from the phloem of plants and directly siphon off carbon-rich compounds. And so that carbon becomes unavailable for the fungal partner. And so we wanted to know whether if we applied aphids and limited the amount of carbon available to the fungal partner, did that change the nature of the relationship? Did the plant change how it was functioning with its mycorrhizal partner? Conversely, to make carbon more available to the fungus, we also applied a high CO2 treatment. So we made it more available to the plant, so more photosynthesis meant that there's probably more carbon available for the plant. And we wanted to look at it using, we had three hypotheses for this work. That when we applied aphids, it meant there'd be less carbon available for the mycorrhizal fungal partner, and that would result in less phosphorus transfer going from the fungus to the plant. We also looked at the high CO2 treatment. We thought, okay, well, if we make more carbon available for the mycorrhizal fungus, then there will be more there'll be more transfer of phosphorus from the fungus to the plant. And when we had aphids and CO2 at the same time, maybe we'd get something in between, because the high CO2 availability would ameliorate some of that impact of the aphids present on the plant. So we used isotope tracers, so radio-labeled phosphorus, which we inject into a compartment within the pot where we have the plant and the mycorrhizal fungi growing. So these compartments are windowed plastic cores, essentially, and the mycorrhizal hyphae can grow through the mesh that covers those windows, but the plant's roots can't. So we create this fungus-only zone within those cores. So when we introduce a radio label into that, only the fungus can get it, and then we can measure how much of it ends up in the plant material. To measure the amount of carbon, what we can do is we release a pulse of 14 CO2 into a sealed gas-tight chamber, and then we measure the opposite. We measure how much carbon, labeled carbon rather, ends up within the core in the fungus, within that mesh windowed core. And because we have paired cores in each pot, we can rotate one of the cores, which severs the connections between the fungus and the plant, or sorry, between the core contents and the plant. So you snap those fungal connections. And so what you can do is look at the amount of radiocarbon present where the connections are intact, and then subtract the amount that has been detected where it's been severed. And that difference is the difference being connected to the plant. So that gives us an idea of how much fungal carbon is present in the scenarios. So what we found, and this is just looking at the carbon, when we had aphids, that's the white bars, and the black bars, or grey bars rather, that's when aphids weren't present. The carbon allocation to mycorrhizal fungi absolutely crashed through the floor when you added aphids, which you might expect because they've got this heavy competition between the aphid and the mycorrhizal fungi. They both want plant carbon, but when the aphids are present, they seem to get the bulk of the carbon and it crashes. There was no effect of CO2. So this is the ambient condition, and then this was the elevated condition. So CO2 didn't seem to make an effect on the allocation of carbon to the symbionts. However, the phosphorus, and this was a really interesting one. So if we look at total phosphorus, this is plant acquired and mycorrhizal acquired phosphorus. You can see when aphids were present, it decreased. However, when we look at the mycorrhizal acquired phosphorus, so this is the radio-labeled phosphorus only, actually aphids didn't seem to make a difference. And so we had this decoupling of the carbon for nutrient exchange when you had aphids in that system. And what this suggests is that in this case, when you add a competitive plant carbon, you change the nature of the relationship. It's not a straightforward one-for-one exchange. So it seems, this is just a quick summary of that experiment, that the mycorrhizal fungi are able to maintain phosphorus supply to their host plant regardless of that host carbon supply in this situation. And actually what happens is that the symbiosis kind of moves along the continuum towards the plant becoming parasitic on the fungus in that certain particular situation. So that really just illustrates the flexibility of the symbiosis, where in normal situations, it might be mutualistic, but different factors can move it along that continuum. And you'll see soon why I'm stressing this so hard. So if we go back to our fossil fungi and we think about, okay, well, they look very similar to mycorrhizal fungi. And I think I don't need to convince anyone that they look really, really similar. And we kind of assume they are mycorrhizas, they're ancient mycorrhizas. But what we can't tell from fossil evidence is what did they do? Where on that symbiotic continuum did they lie? And until we can understand where on the continuum they probably lie, then we can't really make too many inferences about what they were doing in the first place. So a really strong hypothesis for the presence of these ancient fungi in these fossilized plant cells is that they had a role very similar to modern day mycorrhizas in that they helped those ancient plants access mineral nutrients from the skeletal soil or the barely there soil at that time. And in that way, they helped the plants get onto the land in the first place. However, there's no evidence for that just from these structures alone, as I've shown you. It can lie anywhere on that. It could well be that they were parasites. Were they just hitching a lift onto the land with the earliest plants? Were they just not doing anything at all? So some of the questions that we're interested in is what do these ancient mycorrhizas actually do? How helpful were they to these ancient plants? And what does that tell us about the importance of fungi in the development of our terrestrial biosphere? So because we don't have time machines, unfortunately, we have to look at modern plants. And so in order to get an idea as to how they might have functioned in the ancient plants, we look at modern plants that bear the closest similarities to those ancient plants. And so in this situation, we're looking at bryophytes because they're also non-vascular, they don't have roots, and they're structurally much more simple than vascular plants and later diverging lineages. And so lots of people have done some work on this, and what they've found to date is basically in these figures here. So this is looking at fungal symbioses in liverworts, and what we're looking at here is photosynthetic rate, the mass, and the production of gemmae. So gemmae are just like little reproductive structures, like little tiny clonal liverworts. And so produce more of those, it means you're going to reproducing more. And the black bars in this case are the non-mycorrhizal plants, and then you have the mycorrhizal plants are the white symbols in each of the graphs. And essentially what the authors show here is that if you're a mycorrhizal liverwort, you do more photosynthesis, you grow bigger, and you produce more gemmae. Okay, so you seem to do much better if you're mycorrhizal than when you're not. But why was that? So this figure here is taken from a paper just looking at the actual functionality in different plant groups. These two are the liverworts here at the end. And what we see here is that there is carbon for nutrient exchange between liverworts and their fungal partners. And so this kind of builds some evidence. And it's we can see that there is carbon for nutrient exchange between liverworts and their fungal partners, which starts making us think that, yeah, maybe these early fungal symbionts were important in helping those early plants get hold of nutrients and gave them a helping hand onto the land. But not long after that work was done and it was published, this paper came out and it kind of changed everything. It changed the way we started thinking about early mycorrhizal symbioses. So this is Martin Bidar-Tondo and colleagues. They were looking at this liverwort here, which I really like this picture because it kind of makes it look like an ancient plant. It looks a little bit like the Aglaophyton that I showed you earlier with its underground axis and then the fleshy sort of fat green axis underground. And then it has like this little or leafy-like top to it. And basically what they found, so Martin's a molecular ecologist. So he's really interested in fungal diversity and phylogeny. So what Martin likes to do is he identifies what fungus is present in different lineages of land plants and maps them against phylogenies. And what he was trying to do was identify the fungus that was present in the fleshy axis of this plant. And he couldn't do it because it wasn't responding to any of the primers that he was using to test for known fungal markers. And so eventually he found out he used a primer set that had absolutely no chance of it working, but it worked. And what he found was actually that this plant didn't associate with arbuscular mycorrhizas as we thought, which we thought were the ancestral type, so ancient. They're the only ones that form these beneficial partnerships. But he found they actually associated with a totally different fungal clade. So they associated with Mucoromycotina fungi. Okay, and these fungi are much better known for being decomposers. There's also some pathogens in there, but they actually predate, their divergence predates the arbuscular mycorrhizas. So it places them at around the right time to be ancient sort of symbionts. And this liverwort's very special because not only does it look like an ancient plant, but it's actually the most basal clade of the liverworts. It's one of the Haplometriopsida. So you've got a really ancient fungus associating with a very early divergent lineage of liverwort, which kind of starts kind of building this new hypothesis that maybe arbuscular mycorrhizas weren't the symbiotic fungi that helped early plants get onto land. Maybe there were other options. Maybe there were other symbiotic options available to these early plants. Now, Martin came to us then and was like, there's this different fungus here and it could be evolutionarily important, blah, blah, blah. And so my first question, of course, is then going to be, well, that's great, but what does it do? Okay, because I'm really only interested in what things do. And so we decided we were going to do some experiments and find out what this fungus does. Is it mutualistic? Is it a parasite? Or is it somewhere in between? What's the point of it? So we ended up going to the South Island of New Zealand, which was very unfortunate for me. That's where the fungus was, and that's where the liverworts was. So we had to go and get it, which was dreadful. So I went over with Jeff Duckett here. There's Martin, Will Rimington, and there's Grace Hoysted and Sylvia Pressel, who I'm going to talk about some of their work very soon. So these are the liverworts in the wild that we collected. So this is Troibia lacunosa. It's a really, I think it's my favorite liverwort. If you can have a favorite liverwort, I think it's mine. It's very frilly. It's really nice, folded kind of thalli. Very fleshy part that kind of runs along the ground again. And then this is the haplometria, which we showed earlier. So this is all you can see when you find it. You just see like the lettucey tops and all that fat axis is underground. We also collected these guys. This is a terrible picture of Allisonia. And then this is Neohogsonia. So these ones were exciting because they form dual symbioses with both types of fungi. So they have associations with the arbuscular mycorrhizas, like most plants do, but they also have associations with that Mucoromycotina fungus that I was telling you about. And we brought them back to the UK, back to Sheffield, to do some of these experiments. Actually, some of these were in Leeds as well, they were split. But this is the experimental system, and it was based on one that was devised by David Johnson, Jonathan Leake, and David Reed in Sheffield. And it's using these mesh-covered cores that I've already discussed, but we covered the windows with a much finer mesh this time because we wanted to exclude the rhizoids of liverworts, which are much finer than roots, obviously. So we create those fungus-only zones, and we can introduce isotopes and measure how much of that gets transferred from the fungus to the plant and vice versa. And this is kind of what they look like from above in real life. So to do the CO2, we did that again, but we used 14 CO2. I've explained this already, so I'm not going to do it again. But we carried out these experiments at 2 different CO2 concentrations because we wanted to see not only how are they functioning now, but we also kind of wanted to impose a Paleozoic style high CO2 atmosphere on them just to test the plasticity of that relationship. So could we force it along that continuum either way? And what effects, so how might that have functioned if it was 500 million years ago or something? And this is what we found. So I'm going to talk through these graphs quite slowly because they're pretty horrible. I haven't figured out a better way of doing it yet. So we've got on this panel on this side here, we've got the Mucoromycotina associations. That's Haplometrium and then Troibia. The black bars are I think they're the high CO2, and the white bars are the lower CO2. Then in the middle, we've got the arbuscular mycorrhizas, with Pricea and Marcantia being the liverworts that hosted those types of fungi. And then we have dual colonized plants, which is the Allisonia and the Neohogsonia that I just introduced you to a moment ago. And really, the only message I want to get across with this is that firstly, if you look at the Mucoromycotina, yes, particularly this bottom one, if we look at this one, they get carbon from their plant partner. Okay, so those fungi are receiving carbon from their liverwort hosts. And they're receiving quite a lot. It's certainly in the same ballpark as some of the other liverworts. But what's most exciting, I thought at least, was that actually the plants that hosted both fungi together, they actually allocated the greatest amount of carbon at both of the CO2s that we tested to their fungal partners, suggesting it seems to be more expensive to the plant to host multiple fungal partners at the same time, which is kind of interesting. And we'll come to kind of evolutionary implications of that in a moment. So then to look at the opposite direction, how much the liverworts were benefiting from the associations. Same experimental systems, but we used 33P and 15N traces to measure the amount of nutrients going from fungus to plant at both CO2s again. So the top graph is phosphorus and the bottom one is nitrogen. And again, if you look at the Mucoromycotina panel in these, we were getting nutrients being assimilated by the liverwort from the fungal partner. So this is actually the very first evidence that this is full-on mutualism. Okay, so just as mutualistic as liverwort arbuscular mycorrhizal symbioses are, which was really exciting because it's this whole new fungal symbiosis that we didn't know about. The second thing that was really interesting was that it was much less CO2 responsive than the arbuscular symbiosis. But the most exciting thing was, perhaps certainly for phosphorus, was that actually under the lower CO2, so modern day conditions, the plants were benefiting from those symbionts much more than either of the single ones. Okay, so it seemed to be, although it's more expensive, you get more benefit from having 2 fungal partners at the same time in these situations. So this kind of started giving us ideas as to why these plants, why these plants are hosting multiple fungal symbionts right through to present day, right? Because I mean, these are modern plants, and there's clearly an advantage to having more than one fungal partner, even though it's a bit more expensive. Also was interesting to note was this nitrogen effect. We seem to have quite a lot of nitrogen being assimilated by the plant, much more than we expected. And we wondered, maybe there's some sort of complementarity between the types of fungi. Maybe one type is more responsible for phosphorus supply and the other one they're using for nitrogen supply. So we kind of came up with this experiment. Oh, I haven't got the slide. That's a shame. Okay, it's fine. I can explain it. So we wanted to test what the role of this Mucoromycotina fungus was in supplying maybe a different nutrient to plants than we would typically expect with an arbuscular mycorrhizal symbiont. So we came up with an experiment where we wanted to test whether nitrogen was important to the Mucoromycotina symbioses, and secondly, could they access it in an organic form? So typically, typically plants only access inorganic forms of these nutrients, and we wanted to test whether actually, because Mucoromycotina, they have saprotrophic, so they have decaying fungi within the same family, were they also able to decay stuff, unlike arbuscular mycorrhizas? And so we, very similar experimental system, but we created an algal necromass, which we labeled with 15N. So we grew algae in like a labeled solution, which meant it assimilated 15N into its biomass. We then dried it down and we included that in the core. So there was an organic source of nutrients within that core. And then we did that set of experiments at high and low CO2, and we injected 33P as well as having access to that organic nitrogen source. And then this is what we found. So this is phosphorus, and it's not terribly exciting. So where we've got plus algae, that's where we included that organic nutrient source in the core, or we didn't. And it didn't really make a difference whether it was there or not. Everyone got phosphorus. It didn't really matter whether they had access to organic nutrients or not. And it also didn't really matter what the CO2 was doing. It was kind of uniform, which is, it's good, but it's not very exciting. But what was really exciting was actually the nitrogen response. And what we found, so static refers to when the fungal connections between the plant and the nutrient source were kept intact, and then rotated is when they were severed, so they either had access or not. What we find is in the Mucoromycotina plants that had access to that organic patch, we got a much greater assimilation of nitrogen, labeled nitrogen, from the organic matter than the arbuscular mycorrhizas. And that held true in the dual colonizations as well. So that was really exciting. That's kind of evidence of there being some functional complementarity between those two different fungal types, and that actually maybe one type of fungus is responsible for gaining one type of nutrients and the other one is doing something slightly different. That kind of gives us some ideas as to how and why these fungi have persisted across millennia, millions of years even, to be widespread today. So we now know that this Mucoromycotina symbiosis occurs across nearly all land plants. In fact, we also have evidence for it occurring in angiosperms as well. So it's a widespread, globally important symbiosis. But what we don't know is what does it do in vascular plants? Okay, these liverworts, they're non-vascular, they're weird. All right, they're not that interesting to everyone, maybe. I don't know why. Yeah, so we wanted to see what does it do in vascular plants? And so luckily for us, we were able to identify a type of club moss that forms exclusive associations with Mucoromycotina fungi. And this is it. So this is Grace Hoysted's work, who she's just about to take up a lectureship at University College Dublin now. But she did this work while we were back in Leeds. And yeah, so we found this club moss, which is called Lycopodiella inundata, and it forms exclusive Mucoromycotina associations in both the gametophyte and the sporophyte generations of its life cycle. And this is some of the microscopy that Sylvia Pressel at the Natural History Museum has done for us. And what she's done is characterize the intracellular sort of morphology of this fungus in this plant. And you can see very characteristic structures that don't really look exactly like arbuscular mycorrhizas. They look quite different. So you've got these very fine branching hyphae that form these sort of fans inside the cells. So you can see them kind of doing it there. They kind of branch in a very different way. They sort of do this. I can't describe it better than that. And then they also form these like swellings along the hyphae, which we're not sure, are they vesicles or if they're spores? We don't really know yet. But what she found most excitingly, perhaps, is that actually where we were picking out the Lycopodiella, Martin, who did the sequencing of the roots, he found that that same fungus was also colonizing liverworts that we were finding, but also grasses and rushes. Okay, so it was super widespread within that area. And even more excitingly, found that this fungus was actually really closely related to that same fungus we were collecting in New Zealand, which is great for our sort of CO2 footprint. We don't have to go to New Zealand to get the fungus. It's also on Thursley Common. But anyway, so Grace did a bunch of experiments looking at the functionality of that fungus in vascular plants. And she found that, yes, there was carbon allocation. So actually we had two different CO2s again. The 800 was kind of representative of CO2 concentration at the point at which lycophytes diverged on the land plant evolutionary tree. Again, just to test the flexibility of the relationship rather than to see what it literally did at that time. But you can see CO2 didn't really make much difference across the board for all of these different measurements. But most importantly, we found that we were getting carbon going from plant to fungus, and quite a lot of it. We found that the fungus was delivering phosphorus to the plant. And we also found, really excitingly, quite a large amount of nitrogen was being transferred to the plant by this fungus, which was quite unexpected because this is a lot more than we typically see when we do similar things with arbuscular mycorrhizas. So it seems this fungus is important in nitrogen nutrition of plants. And then just to hammer that home, Gerhard Gebauer and his student Philippe Giesemann did some stable isotope work looking at the natural abundance of these stable isotopes in plants and the surrounding neighbors. So the plants that were colonized by Mucoromycotina fungi were the yellow dots in both of these figures. And then the neighboring plants that weren't, so these just had arbuscular mycorrhizas, are the purple dots. And essentially, we're looking at the discrimination on the nitrogen axis here. And you can see these plants separate from Glomeromycotina colonized plants, suggesting that they're getting their nitrogen from a different source than those other plants. Okay, and we're thinking that it's coming through the fungus, and so it's becoming accumulating that heavy isotope in the tissue. So we have this kind of really exciting new symbiosis that we don't really know very much about. We're getting there, but we don't really know much about it. And there was, in 2017, an Australian group linked the fungi that we were studying with what have been long known as fine root endophytes. Okay, so fine root endophytes are a group of fungi that have been we've known about them forever, but they've pretty much been ignored since the 1960s. And that's mostly because of pictures like this, right, where they look a bit like arbuscular mycorrhizas, and they're just a bit finer. And what's happened is that they've been lumped in with arbuscular mycorrhizas. And so all of the classic literature where they're looking at arbuscular mycorrhizas, they just kind of lump fine root endophytes in with that. And so they've even been called, that for a long time they were called Glomus tenue, and Glomus is an arbuscular mycorrhiza genus, right? So we've got this huge confusion in the literature between fine root endophytes and arbuscular mycorrhizas. And they've always been thought to be functioning in the same way as an arbuscular mycorrhiza. However, it seems from our work that they're not actually, they're different. And they've now been renamed to Plantis scortium tenuis for reasons. I don't know why this group kind of did that randomly. But we know that they're closely related to the fungi that we find in our liverworts and our Lycopodiella experiments. So we're kind of trying to bring fine root endophytes back into the spotlight. And I think we're getting there. So we've basically shown that these symbioses are mutualistic, in that they exchange phosphorus and nitrogen for carbon in liverworts, lycophytes. We've also got evidence now in angiosperms. They're CO2 responsive in some groups of plants, but perhaps most importantly, they seem to be everywhere. So they're in Trifolium, Lycopodiella, Juncus, Holcus, Molinia, Fossambronia, even in some lineages that don't seem to be mycorrhizal, they seem to have these fine root endophytes. So we've got this really exciting time now where we're kind of learning more stuff about this diverse lineage of fungi. But I mean, let's take a step back from that and kind of think like, so yeah, great. Well, we know there's lots of different types of mutualistic fungi and they have this role in nutrition. But what does that mean? What does that really mean in terms of development of the Earth and evolution of the biosphere and the atmosphere even? So one of the questions we've been really interested in is how does this, what's the point of having these diverse fungal symbionts, and why should we care about diversity in things that we can't really see? particularly, so I've been working with Ben Mills at the University of Leeds, and he's a geochemical modeler with a real interest in paleoclimate. So he models paleoclimates and how different things influence it. So chatting with him, and he was kind of convincing me that actually fungi might have been important for development of Earth's climate. And I was kind of like, what are you talking about? That's nuts. But I'm going to explain to you how I explained it to me, and hopefully you'll be convinced as well, because I was. But basically, you've got plants with mycorrhizas, okay, and the mycorrhizas help those early plants, which are tiny, like tiny, so they get access to mineral nutrients. And this is a picture here. This is an electron micrograph. You can see a mycorrhizal fungal hypha growing along a soil particle, interacting really closely with it. This is another one, germinating mycorrhizal spore, and you can see the hypha growing from the spore and actually trenching through the mineral. Okay, so it's secreting organic acids and enzymes and stuff that actually are able to dissolve that rock, and you get left behind with this sort of trenched pattern, which I just love. Anyway, so by doing that, they're able to get access to nutrients and pass it to those early plants. This lets those early plants get really big. They start growing into like trees and things with roots and really big, hefty plants, big liverworts. Now, this increase in plant complexity and biomass exerts a much greater drawdown on CO2 in the atmosphere of the globe. And in turn, that kind of drives changing climate. So what we then decided we wanted to do was like, oh, okay, right, okay, well, fine, but we know these fungi operate differently. They do different things. The different groups work differently. So what are the scenarios that could have happened in those different sort of symbiotic scenarios? So Ben, I say we very grandly, Ben modeled it. Okay, so he used the experimental data that I've shown you today, and he modeled it in his in a COPS model, which is just a box model, basically feeds all of that stuff in and comes out with predictions about climate. So this is NPP, net primary productivity, which is kind of a proxy for like photosynthesis, right? How much photosynthesis was going on. The black line is if there were no fungal symbionts at all, and the x-axis goes from 500 million years ago, which is when plants first came onto the land, through to sort of not modern day, but kind of when angiosperms hit the scene. But yeah, okay, so you can see the black line is where if there were no fungal symbionts, just business as normal. If everything only had arbuscular mycorrhizas, we're looking at the blue line in each case, and in this one, you can see it's much greater than if no one had any symbionts. The red line is if everything was only Mucoromycotina fine root endophytes, and you can see it's much slower. MPP didn't really take off anywhere nearly as quickly as if everything was arbuscular mycorrhiza. This then had an effect on the rise of oxygen, either accelerating it in the case of if everything was arbuscular, or actually delaying the rise of oxygen if it was all Mucoromycotina. And then that obviously has impacts on atmospheric CO2 concentration, with it decreasing much more slowly if everything was Mucoromycotina powered versus if it was all arbuscular, which then has big impacts on global temperatures. So according to Ben's model, if we had Mucoromycotina dominated plants, then it would have been much hotter for much longer than if we had arbuscular mycorrhizal symbionts. I mean, obviously, I say obviously, but I mean, the reality is it's probably somewhere in between because everything was mixing up and it was all a bit in between. But I think it kind of illustrates really nicely the impact of these fungal symbionts and how big that impact could have been on the on development of Earth's climate and the rise of the terrestrial biospheres. And essentially, the message I kind of wanted to get across with this was that fungi really mattered in how plant evolution got going, and in how we see the world around us today. And we need to kind of consider fungi when we're thinking about these big global climate change questions. So that kind of brings us into modern day now. And so one of the big projects we've got going at the moment is to really explore the role of this Mucoromycotina fine root endophyte in modern land plants and in terrestrial ecosystems today, because we know nothing about it. Like, we want to know through this new project that we're now doing, we want to know which plants do these fungi actually associate with, where are they doing it, why are they doing it, what are they doing, how are they doing it, and how does environment affect it, so that we can get a better handle on the impacts of these fungi on geochemical cycling. And also, you know, you can't protect what you don't know about. So we're kind of working on that now. So I kind of wanted to bring this then, just in the final few minutes of the talk today, just to go into kind of like applications and looking forwards and why this stuff matters in a practical sense. So we know today that more than 90% of plants have these fungal partners of one type or another. And that includes the stuff that we eat, that includes crops, okay? And I'm sure you've seen all this sort of stuff before, but we're kind of facing this crisis in food security as a result of a combination of factors, including rising CO2, growing populations, and depletion of natural resources. And so one solution that's quite commonly touted is that actually we can use mycorrhizal fungi potentially to help support food production by reducing inputs of chemicals, because they can help crops in fields get access to alternative nutrient sources and yada, yada, yada. And so one of the things that we've been really interested in over the last few years is actually looking at crops and crop mycorrhizas to see how useful are they really, and is it a realistic alternative to applying synthetic fertilizers in agricultural scenarios. So we know wheat and barley, for instance, are very capable of forming mycorrhizal associations. But what's interesting is actually the ability to form functional associations is actually really variable. And that's mostly because of selective breeding for traits such as resistance to disease, responsiveness to nutrients applied. So fertilizers, pesticides, etc., has kind of inadvertently selected against being mycorrhizal. Because you're upping plant defenses, or you're getting them really into sort of a highly disturbed habitat, which isn't conducive to forming good mycorrhizas. And so one of the first things we wanted to do was look at how variable the responsiveness to mycorrhizal colonization was in different modern wheat cultivars. And so this is just kind of more justification for that study. We've got this study I referred to earlier, but here we're looking at vascular plants, and you can see under high CO2 concentrations, we had this big decrease in mycorrhizal function. All right, and that's kind of worrying if we're actually recommending people to be using mycorrhizas in the future, because maybe they're actually going to be a bit rubbish. So we wanted to test that, and we wanted to see how the functionality is likely to change with predicted future climate scenarios. And so this is the work of PhD student Ashley Elliott, and she looked at colonization using a commercially available inoculum. And essentially what she found was that the inoculums worked, that these cultivars, we had 3 different wheat cultivars. We had Skyfall, which is like a really important modern one that's planted very commonly across Europe, Avalon and Cadenza, which are kind of really useful breeding lines. And she found when she inoculated them, which are the white bars, they increased in colonization, right? Which it's not very exciting. But it proved a point. These crops do form mycorrhizas, and they can do it, but it's variable across the cultivars. So some cultivars responded much more readily to colonization. For instance, Skyfall, if you compare it to Cadenza, was much more resistant to forming associations. Tom Thurkel took that a step further, and he actually tested a response to mycorrhization. So he looked at their growth response in plants that had mycorrhizas versus plants that didn't, across this mapping population of wheat. So he tested 100 different lines, and he found that actually you got this whole swathe of different responses to mycorrhization in those crops. So some varieties, they could increase biomass by up to 50%, whereas some actually decreased. All right, and it's probably to do with compatibility between the fungi. So to try and explain that, he had a look at the phosphorus concentration of the tissues, and what he found was in the plants that were not mycorrhizal, as they got bigger, they tended to be reduced in phosphorus concentration in their tissues, suggesting there was this dilution effect in the tissues. So bigger plants, finite amount of phosphorus, it got diluted, so they kind of couldn't make the most of it potentially. But what he found was in the mycorrhizal plants, that relationship was much less strong. So they kind of alleviated that nutrient dilution effect in the tissues, which you can read about in that paper. So then he kind of wanted to look at the actual functionality then. So what proportion of the phosphorus those plants were getting, is it mycorrhizal? So he looked at the same 3 cultivars that Ashley had looked at with the colonization, and he found that total phosphorus was very variable. But when he broke it down into the mycorrhizal phosphorus, he found, again, very variable, but it wasn't the same way as with total phosphorus. So he found that some cultivars accrued a lot of mycorrhizal phosphorus, whereas other cultivars didn't. And he also found there was not this significant CO2 effect within the cultivars, which is a great news story, right? It means that there's still propensity for multi-crop cultivars to make use of mycorrhizas, and that functionality is probably not going to change with changing CO2 in the future. With nitrogen, he found a similar effect. There were differences between cultivars. He did the same thing in barley as well and found a very similar story. Carbon's less interesting, but also very optimistic in that they did transfer carbon to the mycorrhizals, but the amount wasn't affected by the amount of CO2 in the atmosphere. So this kind of is a really good news story for the future, really. Just to summarize those sets of experiments, we found that there was cultivar-specific receptivity to mycorrhizas in wheat and in barley, and that actually that increases with inoculation. We found that mycorrhizas were able to increase wheat nitrogen and phosphorus uptake, but this ability varies according to cultivar, which kind of means we have the potential to exploit this ability in the future, maybe breed for these mycorrhizal responsive traits. We found that mycorrhizal mediated nutrient uptake was not linked to plant carbon outlay. Again, that's a really positive thing if we want to use them in agriculture, because it's not going to cost the plant more in terms of carbon it could be using to feed into its yield or other biomass above ground. And so it seems that the functionality of this symbiosis, particularly in wheat and in barley, is not affected by changing CO2 in the atmosphere. So again, good news for potential use of agricultural mycorrhizas in the future. And so I'm going to finish it on a very positive note because I'm a very positive person. And yeah, so thanks to everyone who actually did all the work. So everyone in my lab, my collaborators and funders who have sponsored us or have provided the funding to let us do that work. So yeah, and that's it. Thank you very much.
Lots of detail, and I'm sure there's lots of questions. So, you got any questions? Or just
No, sorry, there's a lot.
Richard, you were first there.
I was just thinking if the earliest fungi, there wouldn't have been much nitrogen to try to swap out, which suggests that
Mucos, yeah.
that they wouldn't perhaps have been so useful to the earliest land. I don't know if that's been factored in at all. But you think that the kind of fungi that get their stuff from rocks would be useful to the earliest.
No, I think it's a good point, but I think, you know, it kind of, I mean, I'm going to look at Sandy here, right? So like early environments, we think of them as being barren rocks, but in reality, it wasn't like that, right? I mean, there was kind of a biofilms and there was detritus and organic matter building up right from the word go. And I mean, the picture I showed an illustrative photo I'd taken of kind of like algal and cyanobacterial mats that just wash up onto those land surfaces, which is where the first plants would have started and started forming those associations. So I think there was organic matter available. It's just it would not have been available to plants on their own with having no roots or probably no abilities to actually get hold of those nutrients. So they would have needed, and I think there is actually certainly molecular evidence that there were lineages of fungi on terrestrial land surfaces way before plants. So you had sort of saprotrophic stuff going on before. Right, exactly.
The 10% of plants that don't.
Yeah.
So what's up with them?
I know, what is up with them? Yeah. So lots of different reasons. None of them have been completely verified. We don't really know. One group that really sticks out are the mosses because it's an entire clade. Like, and I mean, if you're a moss, surely it makes sense to have a fungal partner because you're tiny, you grow on like rocks. But they don't. They just don't do it. Lots of different reasons why they might not do it. So moss rhizoids are quite different to things like liverwort and hornwort rhizoids and root hairs because they're multicellular. So it could be an access thing. They're also a lot finer, so it could simply be that the fungus can't get in. They also gain, maybe they just don't need it, because I mean, a lot of mosses get their nutrients from rainwater and dust. Maybe they just don't need it. Maybe they're there and we just haven't sequenced them. Maybe, I mean, you know, I think one thing we've learned with all the Mucoromycotina work is that, you know, don't count anything out because, I mean, you've got to be using the right primers to detect stuff, and if you're not detecting it, doesn't mean it's not there.
So for the vascular plant.
Oh God. Yeah.
Just we haven't detected yet.
Oh. Yeah. No, there are vascular plants that seem not to have them. So brassicas. I would have said sedges, but we have detected Mucoromycotina in those plants that we don't consider typically to be symbiotic. So I think I would be very sort of on the fence, and my personal feeling is that I don't think we've detected everything in all plants.
Sorry, I don't know your name next to it.
What asked about the prototaxites?
No.
So, why did these fungi form such large structures? And how did they support the size of the structures?
Oh man, I wish Christine Struluderian was here because she will know the answers to this. I think it's even controversial that they are fungal in nature, right? So there's lots of different hypotheses about what prototaxites is, was, could have been. But the most likely answers, the best we can do is that it probably was a fungal structure, and it was probably a fungal fruiting body. So maybe a huge towering mushroom. I couldn't even begin to answer the rest of that. I have no idea. I don't know if anyone does really. I think it's one of those paleobotanical. Can we call it paleo? Is it botanical if it's fungal? I don't. Anyway, it's a mystery. It's a paleo mystery, I think.
And show how different kinds of cereal varieties vary in their response. How does that tie up with the way they're behaving in a field now? Are the cultivated conditions just working against mycorrhizals, or are they actually responding and being infected and so on?
No, really, yeah, great question. And it's a really interesting sort of, from an evolutionary standpoint, scenario, because what you have is this, in a conventionally managed field, you have regular tillage, so you've got massive disturbance of the soil all the time. You've got massive periods of time essentially when there's very little carbon flowing into a fungal network, if you like. And you have rotation, you have different plants being planted. And so what actually, it's not conducive to a very diverse, long-lived fungal community in the soil. But that doesn't mean they don't exist. So what you find in agricultural soils is that you have a much lower diversity of species, and the species that tend to exist there are very weedy and able to complete their life cycles very quickly. So what's actually happened is not only have we kind of bred crops to do really well in those sorts of conditions, but we've also inadvertently kind of selected the fungal communities in the soils to also work with those conditions. And those fungi are not necessarily the ones that are going to be most beneficial for the crop, because they're going to need to get as much carbon from the plant as possible and complete their life cycle very quickly before that crop is harvested and all that carbon's taken away. And so I mean, there's lots of evidence about diversity being much lower in agricultural fields than in conventional grasslands, for example. But equally, the propensity is there if we change management practices to almost naturally reseed fungal communities in the soil, because spore banks last for a long, long time. They're also wind dispersed, so all of that cultivation, it blows up spores, and so actually you do have a lot of spore mixing, and you can detect mycorrhizal fungal spores everywhere. And so I think it's kind of a matter of management and kind of changing how we manage the soil to kind of try and manage the fungal communities. I think it's going to be a much better tactic than kind of introducing new varieties into the soil, which will just get outcompeted by these very fast weedy things. Yeah, I think there's not a massive amount of evidence that commercial mycorrhizal inocular are much use. Again, it's very context specific, isn't it? It depends what species mix is. It depends on the conditions that you're adding them to. But I think given that mycorrhizae or mycorrhizal fungal spores are certainly highly abundant in almost every soil everywhere, what you're doing, introducing new varieties in, is they're either going to become very dominant very quickly because they're very aggressive and very good at forming associations really quickly, but who knows how beneficial they are. Or they're just going to get outcompeted by what's already there, that's already adapted to the local conditions within the soil that you're adding them to. So yeah, what we found, so what I haven't presented it here, but what we found as part of Ashley's PhD work, where she was using a commercial inoculum in the wheat, was that she had this increase in colonization with the commercial inoculum. So the structures were all there, but when she did the tracing and she was looking at what they actually were doing, not much. So they were there, but they were just loafing. They weren't doing anything. So again, it's one of those difficult things where things have been sold without full story being kind of got at, I think, which is kind of, it's not to say that it's not of use, because I think in certain situations it would be, and I think that there is potential to use it to our advantage in producing food or, you know, conserving species or gardens or whatever. But I think it would need to be a much more nuanced, tailored approach to actually get the most out of it. And sometimes working with what's already there is the best way anyway.
Can we have what? There was one question at the back there. So is the last one. Whoever was at the back. Yes.
Yeah. So exploring what you were saying about sustainable agriculture, is there a future in genetic modification for expanding this to improve the relationship between crops and nutrient exchange, particularly in nutrient-deprived areas?
Yeah, great question. Lots of factors to consider in answering that, though, because it's not just about can we do it, there's a whole should we, is it acceptable to do it? If we're going to modify, if we're going to do genetic modification of crops, there's that whole debate. But putting that aside, Tom's work looking at the mapping population, it certainly suggests we can breed for it because it is linked to traits. He did the QTL analysis, so there is potential you can breed to improve certainly responsiveness to fungal colonization. I don't know if you can. I don't think it's been identified. I don't think. Well, maybe we can. I mean, there's certainly groups working on improving mycorrhization in crops, receptivity. But I think we haven't. I don't think we know. I don't think we know if that actually works. Certainly, you can increase colonization, but as I just referred to then, colonization doesn't always mean very much if it's not actually functioning. And I imagine it's a very complex genetic toolkit that kind of governs functionality. You can tell I'm kind of way out of my wheelhouse here. I think probably most likely is breeding. I think you can go down the breeding route with it, and it certainly is linked with traits that you can breed for. So that would be the route I would go for. Yeah, so they start, so the plant seedling roots start sending out sort of symbiotic signals very early in development. And you can typically have good colonization between 2 and 6 weeks, kind of they colonize. But again, it depends on how many spores are in the soil, how many roots are being produced,
One last question at the big point here.
Oh, I was just wondering, how do you, I think you said that most club muscles don't have mycorrhizal.
Most what, sorry?
Most of the club muscles don't have mycorrhizal.
Club mosses.
Is that what you said?
Mosses don't.
I thought you had a program where you said that one club muscle tribe.
Oh, I see. Sorry, beg your pardon. Yes. So
I was wondering how you go about identifying the one.
lucky. No, so club mosses do form associations, but mostly we've found previously with using what I call the old set of fungal primers for identifying fungal symbionts, arbuscular mycorrhizas. And it was part of the big screen that Martin was doing, looking for Mucoromycotina, that he picked out a club moss that we'd seen with microscopy, that it had colonization, but it hadn't picked up with the form primers. And so he honed in on that one for that experiment in particular. But I don't know. I don't know how broadly we're looking at other club mosses, I guess, as part of our big screening project. But yeah, no, they do form them. Some don't, but I mean, There's always the odd one.
Yeah, that's great. We'll have to finish now. And come and ask the speaker separately
Mhm.
later. So thank you very much once again. That was
Thank you.
wonderful. And thank you very